All right, so in this session, we'll now look at smooth muscle. So smooth muscle, we're saying this is the second most abundant type of muscle in the body. Second most abundant type of muscle in the body. Aside from that, all smooth muscles are involuntary muscles, meaning they are controlled by the subconscious brain. Therefore, their action is not controlled by our awareness. Then, in terms of their attachment, like we looked at, smooth skeletal muscle, the attachment is skeleton. Then cardiac muscles, we said their attachment is endothelium of the heart. But when it comes to smooth muscle, their attachment is in hollow tubular and cavities of the body. For example, tubes. We're talking about ureters, urethra, fallopian tubes, vas deferens, urethra. Hollow organs, we're talking about, for example, urinary bladder, stomach. We have various other cavities in the body, chest cavity, abdominal cavity. Then sinuses equally have smooth muscles in them. Then all blood vessels are supported by smooth muscles. So blood vessels are part of the tubes that we have in the body. Even bronchioles, bronchi, trachea, all those have smooth muscles around them, and their contraction and relaxation is controlled by the subconscious brain. So in terms of function, smooth muscles are there for peristalsis. So this is onward propagation of contents of tubes and hollow structures of the body. For example, the digestive system, we have cavities like the stomach. Then we have tubes like the small, large intestine, esophagus. All those undergo peristalsis. Ureters undergo peristalsis and move urine towards the bladder. Regulate blood vessel diameter. So the smooth muscles in the blood vessels are involved in expansion and contraction of blood vessels. If there is sustained contraction of blood vessels or inability of blood vessels to expand in response to the diastolic phase of the heart, the systolic phase of the heart, we find that now the blood pressure increases. Regulates bronchial diameter. So bronchioles are also regulated by the smooth muscles in them. So the diameter of the bronchioles is influenced by those smooth muscles. In patients with asthma, we find that because of various triggers, for example, dust, coldness, pollen, even drugs, these smooth muscles undergo rhythmic contraction and relaxation, what we call spasms, and cause occlusion, because these bronchioles now are not fully open, and therefore air reaching the alveoli is limited. Pupil aggression here. So the pupil dilates and constricts in response to light. When there's too much light, the pupil constricts. In dim light, the pupil dilates so that more light can enter in the eye. Okay. So those are the major general functions of smooth muscles. Come in. Come in. Mm-hmm. Yes, we're having class at 12.
What's day?
Because 10 with a different class. You're coming from far?
From far. You didn't say anything.
No.
This was anything.
I was just talking to him because I want us to meet physically.
No.
Yeah. So wait at 12 hours. If I finish early, we may start early. So just hang around. Okay. So shape of smooth muscle, we are saying they are elongated cells with tapered ends. So the ends are narrow, like we can see here. They are elongated, they are long. Then the ends are narrow. Look at that. So this is a contracted muscle. That is a relaxed, smooth muscle. It's long. Here it is reduced in length because it has undergone contraction. Then they have single nucleus for them, and the nucleus occupies the central part. So the nucleus is always in the mid portion here. That's where the nucleus is. In terms of organization, similar to what we saw with skeletal muscle, we have endomysium covering individual smooth muscle cells. Okay, then we have perimysium covering a fascicle, then epimysium covering the whole muscle group. Now, we're saying when the muscle contracts, the nucleus becomes distorted in shape. The catchword is that it appears like a corkscrew. This is a screw that is used to open wine bottles. So it's more like a spiral, so which is shown here. This is a relaxed muscle, that is a contracted muscle. So if you look at the nucleus, compare with this one. Contracted nucleus, relaxed nucleus. So the cell tends to fold on itself when it contracts. Okay. So here, what we are seeing as nuclei of smooth muscles, they are elongated like that. So the cell borders are not visible. What we see are just the nucleus. So all these are nucleus. Even these are nucleus. So these are circular cells. So they are going around the tube like that in a circular manner. So they are going in this fashion. Then these are longitudinal. So these are going vertical. So we are looking at them from above. These are longitudinal. That's why it says outer longitudinal, inner circular. So this was cut from the small intestine where we have such kind of arrangement. The inner smooth muscles are circular, going around the tube. The outer muscles are longitudinal, following the long axis of the tube. Okay. So in terms of organelles, smooth muscle have similar organelles like any other cell. They have mitochondria, they have numerous ribosomes, rough endoplasmic reticulum, Golgi apparatus. So here, the rough endoplasmic reticulum present Golgi apparatus. Of course, these are there for protein synthesis, carbohydrate synthesis. Protein is rough, carbohydrate, Golgi. Okay. Now, on the cell membrane, remember when we're discussing skeletal muscle, we said on the cell membrane where the nerve cell comes close to the individual muscle cells, there are invaginations or folds on the sarcolemma, which is a cell membrane of muscle cell. Here they are called calveoli. Okay. There we're calling them T-tubules. So they are short on smooth muscle. They are not as long as the T-tubules. That's why the name is even different. We call them calveoli. Okay, but that's where interaction occurs between the nerve and the muscle. There's always a small space, what we call the neuromuscular junctions. Okay. So when it comes to organization, smooth muscle, yes, have actin and myosin, but these are poorly organized, not forming sarcomeres. So we don't have sarcomeres in smooth muscle, like we saw in skeletal and cardiac muscles, but actin and myosin is present. And since these are the two proteins that are involved in muscle contraction, even here they will produce contraction. So let's look at that. How are these arranged in smooth muscle? So I'll take us back to this diagram. So this is smooth muscle there. What we have is actin and myosin. Remember this arrangement on a sarcomere. Similar, right? Sarcomere was from one Z disc to the next Z disc. Now here we don't have Z disc. Instead, Z disc is replaced by dense bodies. You see those? Those are dense bodies made up of micro tubules. These are microtubules forming dense bodies. So the two dense bodies act as point of attachment for myosin and actin. So this is equivalent. What we are seeing here, this structure is equivalent to a sarcomere, because in a sarcomere, where there's a dense body, there was a Z disc and a Z disc here. Then we have this arrangement inside. Now, what happens is one dense body is attached to the nucleus. So the whole nucleus on its surface here, it has dense bodies. So think of this point here as being on the nucleus. Then the other dense body is found on the cell membrane, the inner aspect of the cell membrane, here inside the cell membrane. So what we are seeing there are dense bodies, dense bodies, dense bodies. So one dense body on the inner layer of the cell membrane, the other dense body on the nucleus. So you're seeing those connections there, from the dense body to the nucleus. The cell membrane, nucleus. Cell membrane, nucleus. So what happens is when the two thick and thin filaments, or myosin and actin, slide past each other, the process is similar. What happens? First of all, troponin has to be activated. Tropomyosin has to be activated. Then actin is activated. Then actin attaches to myosin. Then the heads are pulled by the tails. I would refer you to skeletal muscle, where I explained in detail what occurs for these two filaments to slide past each other. So when that sliding occurs, this distance here between the two dense bodies is shortened, because the heads on this side are pulled inwards. So actin is pulled there. Equally on the other side, actin is pulled inside. So what happens is the cell membrane is pulled towards the nucleus. Now, since we have dense bodies all over the cell membrane and dense bodies on the nucleus, when these contract, what happens is the cell shortens. You see that? This is a relaxed muscle. That is a contracted muscle. So this is a muscle that is undergoing contraction. Now look at the cell membrane. This is a contracted muscle. Compare with this one. Here, muscle is smooth on the surface. But here, the muscle has folds, because the cell membrane is pulled inwards by the actin and myosin filaments. So in terms of contraction, it's the same. Actin and myosin have to slide past each other like that. But the arrangement of the contractile apparatus is different. Cardiac and skeletal, we had sarcomeres. Here, what we have are dense bodies plus actin and myosin forming the contractile apparatus. Now, because of this arrangement, we see that the contraction produced by smooth muscles is weak as compared to the contraction produced by cardiac muscles and skeletal muscles. The major difference is this: contraction produced by smooth muscle is sustained over a longer period of time. So the muscle can stay in this contracted phase for a longer period of time without entering what is known as muscle fatigue. So skeletal muscles easily undergo fatigue as compared to smooth muscles. Cardiac muscles, there is no fatigue. The moment the heart is formed, they continue contracting and relaxing for life. So they don't undergo fatigue. But when you compare smooth to skeletal, these can give us a sustained contraction, but that contraction is weak. It can't generate a huge force that can move, for example, a limb. Okay. I hope you have gotten the difference between skeletal and cardiac, skeletal and smooth muscle. All right. So here we're saying the actin filaments lack troponin, so instead they have calmodulin. So when calmodulin acts with calcium, which is produced by the smooth endoplasmic reticulum, that's when actin and myosin attach to one another here. So you're saying they don't have troponin, instead they have calmodulin. Remember troponin in skeletal muscle? Again, I'm referring you to that video where troponin was demonstrated, so that we get the difference. All right. So this is already explained. Now we look at fascia. So we are done with muscles. We've covered smooth muscle. We've covered cardiac muscle. We have also covered skeletal muscle. What I want you to know is, how are the muscles differing from each other? We begin with shape. Look at the number of nucleus. Look at the way the muscle contracts. Can I Then look at the contractile apparatus also. We said in cardiac muscle and skeletal muscle, we have sarcomeres. But in smooth muscle, we have dense bodies and actin and myosin filaments. Both skeletal and cardiac muscles have troponin, which is the first molecule to interact with calcium. But in smooth muscle, we have camudlin. So camudlin replaces troponin. Okay, once you are done with that, next thing is just to know, where can you find these muscles? Where do we find smooth muscles? Where do we find cardiac muscles? And give examples of where you can find skeletal muscles. Once that is done, you're safe to answer questions on these tissues. So last thing now to look at today is fascia. So fascia is simply connective tissue. Now, when we discussed connective tissue, we said connective tissue proper is categorized into two. We have dense and loose connective tissue. So fascia is both. We have fascia that is dense connective tissue. We also have fascia that is loose connective tissue. So what is fascia? Think of them as layers underneath the skin. So when you have the skin, when you remove the skin, what you see next is fascia. Those of you who have skinned a goat before, or even cow, pig, we rarely skin, we just remove the fur on the surface of the skin. But you're skinning a goat, when you cut through the skin, after that, you can separate the skin using your bare hands. You just put your hands in between the skin and that white part that appears. That white part that appears is what we call fascia. Even human beings have that. Now it appears as though it is one layer, but actually it is two layers. So we have a superficial layer and a deeper layer. So after skin, before you see muscles, you're going to find two layers of fascia. Superficial fascia, then you go to deep fascia. The superficial fascia okay, no pressure. Okay.
You need to be part of the community. Almost there. Just be part of it.
Okay. Okay, so that was deep fascia and superficial fascia. So when you're cutting through the skin, the first layer of fascia that you come through after cutting through the skin is superficial fascia, then followed by deep fascia. Remember, it is a whitish membrane that appears when you cut the skin. Call it fascia. So let's look at them and where they are located. So let's start with superficial fascia. So superficial fascia lies just beneath the skin. So once you remove the skin, you enter into superficial fascia. It's white. Sometimes when you cut yourself with a laser blade on the skin, you see white, color there. That is superficial fascia that you're looking at. It's always loose connective tissue. And we covered what loose connective tissue is. This is a connective tissue that has the highest number of blood vessels in it and is found in almost all parts of the body. Okay. What is the function there? So it allows the skin to slide over the muscles. So if you hold the skin can move forward and back because it's not direct in contact with the deeper tissues there, the muscles or the bones. Skin can move a bit. Next is it allows blood vessels to reach the skin as well as the deeper structures. Okay, so blood vessels are coming from the arteries. Arteries are always deep. Veins are the ones that are on the surface. So even the skin needs arteries. So arteries will pass through the two layers of fascia, the deep, the superficial, then reach the skin. So it acts as a conduit or medium through which blood vessels will reach the skin. Last but not the least, since it is loose connective tissue, it has a lot of adipocytes in it. And these adipocytes, we said, are there as fat reserves. And when fat is broken down, it gives us energy. Now, of specific areas where superficial fascia is very common is the lower abdomen, just below the umbilicus there. There is accumulation of fat in that area. So this fascia, superficial fascia in that area, accumulates fat. This is seen in females mainly, but even males. Okay. Worse if there's high intake of alcohol and high intake of fatty food. Where does the body take the fat? It will store it in fascia so that it can use it. So usually fat in the body is unused energy that has not yet been used. And we're saying having a lot of unused energy in the body is unhealthy. Okay, because that may begin to cause disease. Okay, so you see that the belly is a reserve of fat that is below the umbilicus there. And it has to be bent by exercises to lose that fat. So in that region, yes, it is superficial fascia, but it's given a special name as Camper's fascia. C-A-M-P-E-R. Camper's fascia. Then deep to it now will be deep fascia. So this is the second layer or the deepermost layer. So this is always next to the muscles. So all muscles in the body are covered in more like a wrap. And that wrapping is deep fascia. Now the difference is that superficial fascia was loose connective tissue. Deep fascia is dense connective tissue. We said this is strong connective tissue because muscles are contracting and relaxing. So to avoid friction between muscles and the skin, there is this covering around muscles, which we call deep fascia. Now, this deep fascia also helps to compartmentalize muscles. For example, muscles in the thigh. We have muscles that are in the anterior part of the thigh. We also have muscles in the medial part of the thigh. We also have muscles in the posterior part of the thigh. These muscles are separated. You can see a clear demarcation. And that demarcation is called intermuscular septum. So intermuscular septum is an extension of deep fascia. So when the muscle is covered, some of these fascia go in between muscles to separate muscles and put them in various compartments. And therefore, it's known as intermuscular septum. Okay. Then in other areas like the neck, the chest region, is called investing fascia, because apart from putting muscles in two compartments, this same septum goes all the way to the bone. And in that process, you find that it will be covering blood vessels with nerves, more like in a package, muscles alone in a package, then goes all the way to the bone. So in those areas like in the neck, we call it investing fascia, because it begins superficially, but it will go all the way into the deeper structures of the neck and separate muscles from different groups of muscles. Okay. Now we're saying near some joints, the deep fascia thickens and forms what we call the fascia retinacula. Okay. For example, in the wrist, on the wrist here, if you're looking at the wrist, if you are facing at the palm, now looking at the wrist there, there's fascia there which we call flexor retinaculum. Then at the back of the wrist, there is extensor retinaculum. So it holds the tendons of muscles as they are passing from the forearm into the palm, so that these tendons are not loose as we are flexing and extending our arm. So there is the same structure, but the function may be different. Instead of separating muscles and the rest, the deep fascia is forming more like a cuff around tendons, so that these tendons are not out of position. Therefore, we call it retinaculum. If it's one, if it's plural, we call them retinacular. You find a similar structure in the ankles, all there to hold tendons. Think of it like a strapping. Okay, like you have put bandage and you add strapping on top. That's what a retinaculum is. It's simply a strap around joints. Okay. Then lastly, we'll look at the tendon. Come in. All right, so tendon is our last structure to talk about now. So tendon, again, we're saying this is still dense connective tissue. Dense connective tissue forms tendons. Tendons connect muscles to bone. So the point where the tendon, where the muscle becomes a tendon, we call it musculotendinous junction. Okay. And they explained how these tendons are formed. It's because of the coverings found around muscles, the epimysium, perimysium, and endomysium. When those structures leave the muscle, they form tendons. Aponeurosis. Aponeurosis, they are flat. Tendons are round. What is the function of tendons? So number one is that they transmit force generated by muscles onto the structure which muscles are attached to, mainly the skeleton. So tendons transmit the force of muscle contraction to the skeleton, and that's why the skeleton is able to move in a certain direction with regards to which muscles are contracting. Look at shape and size. So tendons vary in shape and size. Okay, we have small, we have large, we have medium size tendons. All that depends on the activity of the muscle. If it's a large muscle having so much force on the bone, then the tendon will be large. Okay. So the role that the muscle plays in the body determines the shape and size of the tendon. Okay, so muscle of great power and force have short and wider tendon. Muscles that control fine movement, for example, muscles in the arms here, these have long tendons. You find the muscle ends before the wrist, but the tendon goes all the way to the tip of the finger there. And if you try to move your fingers and look at your wrist, you see some bulging, more like ropes under the skin. Those are tendons, but the muscle has remained in the arm. So these muscles in the arm are there to control fine movement, like writing, knitting, drawing, typing. such kind of thing. But muscles, for example, in the legs, you find that they have short tendons and wider tendons because they are there to work against gravity and move the whole organism. Okay. What we have here is the muscles of the calf, known as gastrocnemius muscle. It's one muscle, but it splits into a medial head and lateral head. This is medial head. This is medial side. That is lateral head. But it is one muscle. Here it merges to form one muscle. But you see that the muscle ends somewhere here. The rest of it is a tendon. It is shown here, that tendon, going all the way to the heel of the foot, which you know as the calcaneum. This is a calcaneum. Calcaneum. Then that tendon there going into this muscle. So this tendon is known as calcaneo, gastrocalcaneo tendon, or otherwise known as Achilles tendon. So Achilles tendon is the largest and strongest tendon we have in the body. Achilles tendon here. From the Greek myth, the Greeks have written about Achilles, or Hercules if you like. Now, we don't know whether Achilles was a human being or it was just someone's imaginations. But anyway, the story is that the tendon in that individual was cut because he was a fearsome man, used to win a lot of battles and the rest. But when he was captured, the tendon on his leg was cut. And that's where the name comes from, Achilles tendon, something like that. So that's the largest tendon and strongest tendon in the body. Not longest, no. Largest and strongest. So what you're looking at here is a tendon similar to dense regular connective tissue. After all, a tendon is dense regular connective tissue. So what you're seeing here, these are fibroblasts. Now, since it is tendon, the name changes to tenocytes. So those are tenocytes here. So tenoblasts are immature, tenocytes are mature. And you know that it's immature cells that are actively synthesizing the collagen that we are seeing here. Then they mature as tenocytes. So this is a tendon, and the tendon is also having fascicles, bundles of tendon fibers. Then each fascicle has individual fibers there. Then within fibers, we have collagen myofibrils, more like a muscle. Okay, so that's a tendon. Otherwise, this is simply dense regular connective tissue. Okay, how do we divide tendons? So we have proximal tendons and distal tendons. So proximal tendon is at the origin, like here. This is proximal tendon. Then it's inserting here. So this becomes a distal tendon. Otherwise, it's just one tendon, like that. So proximal is from the muscle. Distal is into the bone, distal tendon. Okay. So this is explained already. Tenoblasts are immature. Tenocytes are the mature ones. Okay. And the rest is just for your own study. Otherwise, we don't ask about this, but you can read through for your own information. The nerve endings found in muscles and the rest, I think this was explained under nerve tissue, that we have myelinated axons and unmyelinated axons. So the myelinated ones are called mechanoreceptors, or simply Golgi tendons. So these are there to sense tension and pressure in muscles. Then the unmyelinated nerve endings or axons found in muscles. So these close to muscles, they sense and transmit pain. Okay, that's why you can feel pain in the muscles after running or exercising for a very long period of time or doing aggressive exercises. You will be in pain the following day. Okay, then pressure is because of the unmyelinated one, which we call Golgi tendons. So these are the Golgi tendon. Individual muscle cells covered, of course, with endomysium. Then within that, a nerve comes in and then sends its branches there. Okay. So these are there to sense pressure. Then they are there to sense pain. Okay. So that's how our nerves interact with muscles. Otherwise, we end here for muscle tissue. Next, we look at systems.